So this is the outline of the presentation. Don't worry if you don't understand a word here. You should worry if at the end of my seminar you don't understand a word, because this slide will be presented again at the end. So that means that I was not able to tell you what I'm doing, not that you are not smart. So I will talk about the endoplasmic reticulum, an intracellular compartment, and then what happens in the endoplasmic reticulum, so protein folding, protein degradation. And then I will talk about a novel concept, which is the regulation of the degradation activity in the endoplasmic reticulum, and I will show you how a virus hijacks this mechanism to replicate in our cells, the coronavirus. And then I will talk at the end about a protein in the endoplasmic reticulum that is important for what I will tell you. So let's start from the endoplasmic reticulum. It's very easy. If you have a cell here, you have this compartment and you have black spots on the membrane. These black spots are ribosomes, and they are synthesizing proteins and putting proteins in the lumen of the endoplasmic reticulum. And the lumen of the endoplasmic reticulum is filled with molecular chaperones and folding enzymes and quality control factors that check the protein that arrive in there and assist their maturation. So about 30% of the human proteome is synthesized in the endoplasmic reticulum, and then once the native structure is attained, the proteins are transported at their site of activity. So the proteins that are synthesized in these compartments are all the proteins that have to leave the cell. So for example, antibody molecules, hormones, they are synthesized in the lumen of the endoplasmic reticulum. They attain the native structure. In the case of antibody molecule, for example, you have to assemble heavy and light chains, and you need two heavy and two light chains together. So once the full molecule is assembled, it is transported and ejected from the cell. Also are expressed in this compartment all proteins that are active, or the vast majority of proteins that are active at the cell membrane, channels, transporters, receptors. Also all proteins that are active in the secretory and in the endocytic pathway compartments, for example, Golgi protein or lysosomal proteins, are all expressed in the endoplasmic reticulum and then transported there. And we are studying what happens in there. Protein synthesis. So everything starts with a ribosome that is reading a messenger, and in some cases the protein that is produced has a very short, about 20 residue long hydrophobic sequence. This is called the signal sequence. And once this signal sequence emerges from the ribosome, everything is blocked. It's blocked until the ribosome finds a channel in the membrane of the endoplasmic reticulum. Then synthesis restarts and the protein is injected in the lumen of the endoplasmic reticulum. Most of the proteins that enter the endoplasmic reticulum are modified. There is addition of sugars. So there is a pre-assembled oligosaccharide bound to a lipid in the endoplasmic reticulum membrane, which is transferred by oligosaccharyl transferase on the nascent chain. And is transferred on the nascent chain every time that you have a signal sequence, asparagine, any amino acid, serine or threonine. When such a sequence appears in the lumen, there is transfer of a sugar. And the sugar is very complex. It's formed by 3 glucose residues in red, 9 mannoses, and 2 acetylglucosamines. And this sugar is hugely important. The first thing that it does is that it adds hydrophilicity to the molecule. So when the protein is not yet folded, it is likely to expose hydrophobic patches, could form aggregates. So by putting this highly hydrophilic moiety, you help the solubilization of these proteins. Then once the protein is folded, the hydrophobic patches are buried inside, they are not exposed. But at the beginning of the life of a protein, it's very nice to have some hydrophilic molecule at the surface. And then the second very important task of this modification is that it determines the fate of the polypeptide chain. It determines whether the polypeptide chain will be folded or not folded. It determines whether the polypeptide chain will be degraded or not degraded. And this is explained in the next few slides. So rapidly, after addition of the sugar, two enzymes, the glucosidase 1 and the glucosidase 2, removes the two outermost glucose residues, so that you have a monoglucosylated sugar appended to a protein. And this monoglucosylated sugar recruits folding enzymes and chaperones. For example, calnexin, calreticulin, and ERP57. And these are the proteins that work on the newly synthesized polypeptide to help maturation, the attainment of the native conformation. The protein is eventually released from the chaperone complexes, and the sugar that mediated the association with the chaperones is removed by glucosidase 2. If the protein is native, it can be transported, secreted through the Golgi, and then secreted from the cell, for example. Not always, when a protein is released from the chaperone system, it is native. In some cases, the folding has not been completed. And there is a quality control sensor, a protein called UGT1. This UGT1 scans the structure of the polypeptide chain. And if it understands, if it thinks that the structure is not correct, then this enzyme puts back a glucose here. And if you put back a glucose, you have a signal that again recruits the folding machinery. So the folding machinery arrives again and starts again working on this protein. And then the protein is released again. And then this time it worked. It's native, can be secreted. It may happen that this protein never attained the native structure. This may happen, for example, if you have a mutation or a deletion in the polypeptide sequence. These mutated proteins never attain the correct structure. So the risk is that you end up in a situation where you have retention forever of a protein that cannot be secreted because it's not native. So UGT will constantly put back a glucose residue and recall the protein in the chaperone machinery. Then the glucosidase 2 will remove the sugar, release it, it's not native, UGT1 understands it's not native, puts back again. So this could go on forever. There is ER-associated degradation, luckily. So cells are smart in some way. They don't want to be blocked in a situation where you have a non-native protein, a mutated protein that you cannot fold correctly. So you have to get rid of that. So how to get rid of that? Well, you simply remove a few sugars, for example. If you start removing, so if I tell you that this is a quality control that understands that the protein is not native, and then it adds a glucose here, well, if you remove this mannose, you cannot put back the glucose, for example. So this is a way to prevent futile cycles of binding and release from chaperones. So there are mannosidases in the lumen of the endoplasmic reticulum that at some point intervene and say, well, this protein is here, we cannot get rid of that, we should eliminate, we should really put that outside, we should degrade that. So they start to remove one by one the mannose residues. And this recruits lectins that see that the protein has a reduced number of sugars. Instead of having 9, they have only 5, for example. So this lectin binds proteins exposing smaller sugars, and they help the translocation of this protein across the ER membrane into the cytosol, and then the proteasome will degrade this protein. There is a problem. The problem is that when a protein enters into the endoplasmic reticulum, by definition, is not native. It has to be folded. So in the same compartment, you have a machinery that would like to take this and fold it, make it native so that you can secrete. But in the same compartment, you have a few guys that would like to take this non-native protein outside, destroy it, because these guys understand that this is not mature. It has maybe not enough time to mature. If the concentration of the chaperones is too high, the risk is that you trap this before actually attaining the native structure and destroy that. And this is linked to diseases. For example, in the cystic fibrosis, you have a very complex protein synthesized in the endoplasmic reticulum, multi-spanning protein, 1,500 residues. And there is, in the most frequent case of cystic fibrosis, there is just one residue which is missing, a phenylalanine at position 506. The paradox is that the protein without this phenylalanine is fully functional, but the quality control system of the endoplasmic reticulum understands that there is something wrong. One amino acid is missing. We don't know exactly why the quality control machinery think that this protein is wrong. It's active. If quality control would be based on measuring activity, this protein is perfectly fine. Unfortunately, quality control is based on the architecture. There is something wrong. So this protein is captured by the ERAD machinery and it is destroyed. This protein would need more time to fold. The folding is not completely compromised, but is delayed compared to the wild-type protein. So this machinery intervenes too fast and destroys a protein that, if we would leave more time, would be secreted. And there are other examples of misfolding diseases in which you have a mutation in the protein that delay folding, so that you have degradation before maturation. And there are other diseases, for example, tumors that acquire a high metastatic potential because some component of the ERAD machinery are upregulated. So in an endoplasmic reticulum where you have too much GP78, for example, GP78 recognizes a specific protein before it can attain the native structure, and the protein is called Chi1 and is a tumor suppressor. So if you have a tumor with too much GP78, this tumor becomes metastatic because Chi1 is degraded. So this is to tell you that the activities of the folding and the ERAD machinery, the folding and the degradation machinery, has to be perfectly balanced in cells at steady state in order to have maximal production of protein, to prevent premature recognition of non-native proteins and their unwanted destruction. So this is what we are studying, how the balance between folding and degradation is maintained in the endoplasmic reticulum. And this is the concept of ERAD tuning. So as I told you, at steady state, so in normal cells, physiologic state, the relative activity of this machinery and of this machinery, it has to be adjusted. You don't have to have too much ERAD power, otherwise you trap not yet folded proteins and you destroy that prematurely. What we have noticed, and also other groups have noticed, is that if you look at these chaperones, you pulse cells with radioactivity, and then you remove the radioactivity, and then you isolate samples from your cells after 10 minutes, after 1 hour, after 2 hours, and then out of these samples you precipitate these chaperones, for example, calnexin. Well, the amount of calnexin 10 minutes after synthesis is like this, is 100. After 1 hour is 100. After 2 hours is 100. After 6 hours is 100. So that means that these chaperones, all of them, are very stable proteins. By definition, molecular chaperones and folding enzymes are very stable proteins, half-life 12 hours or longer. But then we have studied, are really all ER resident proteins so stable? When we are studying, when we have looked at these proteins, we have noticed that they are very unstable. So it's kind of, you have two families in the ER. One family, very stable, and another family that is degraded rapidly after synthesis. So the half-life of this protein, EDEM, Yarmanosidase, cell, OS9, ERP, and GP78, ranges between the 1 hour, 2 hours. Rapidly removed. So you synthesize, but then you remove that. So out of this, we made a kind of model saying, oh, maybe this is what happens at steady state in cells. So normally, if there is no problem, the protein that enters into the endoplasmic reticulum should be folded correctly and transported at the surface. So at steady state condition, you have high concentration of these very stable proteins that assist maturation of the newly synthesized polypeptides. And then you have you are trying to get rid of these potentially dangerous chaperones by eliminating them fast. Then, If you have problems in the endoplasmic reticulum, for example, synthesis of mutated proteins that you have to be degraded, then you stabilize this protein, or you induce their synthesis. You enhance the level of degradation. But at steady state condition, the situation is likely to be this one. Very stable, very unstable. So obviously, we were interested in understanding how. So all these proteins are initially in the endoplasmic reticulum. These are ER chaperones. But how can the cell distinguish between these two populations and selectively take these ones and eliminate them? So the mechanism of degradation, what's that? We selected one model protein, which is EDEM1, is one of the proteins that remove the mannoses from the oligosaccharide. Very unstable, rapidly degraded. After 1.5 hours, you don't have the protein anymore. If you put chloroquine, which is a lysosomotropic agent, you stabilize the protein. So this protein is degraded in an acidic compartment, let's say. You have the same result if you treat the cells with inhibitors of lysosomal proteases. So we kind of assume that this protein is transported from the ER into acidic organelles and then is destroyed. How can that happen? Well, for a protein of the endoplasmic reticulum, the conventional way to arrive at the lysosomes is secretion. So you are transported to the Golgi, and then you are deviated into lysosomes, and there you are degraded. Another mechanism to arrive in an acidic compartment is autophagy. So part of the endoplasmic reticulum containing EDEM, but not calnexin, for example, which is very stable, part of the endoplasmic reticulum is maybe eaten up by autophagy. So which one of the two is the correct one? So secretion is not correct. We know that EDEM never travels through the Golgi. And we know that for sure because EDEM is never in the Golgi. And then if it would go through the Golgi, the protein would be modified and would change the molecular weight, something that never happens for EDEM. So EDEM is not transported at the endosomes through the Golgi complex via secretion. So maybe autophagy. So this could tell us that autophagy is really indeed, because autophagy is inhibited if you change the pH in the lysosomal compartment. You inhibit autophagy, you would inhibit degradation of EDEM. However, when we tested the half-life of EDEM in cells wild type and in cells that do not have autophagy because an essential autophagy component is missing, the degradation is exactly the same. So we can eliminate autophagy, but degradation progresses. So it's not autophagy. And more than that, if we enhance autophagy, activate autophagy, for example, by giving rapamycin or by starving cells, we enhance the activity of autophagy, but degradation of EDEM is blocked. So it's for sure not autophagy. If it would be autophagy, when you activate autophagy, you have more degradation. We don't have that. So it's not autophagy, and it's not secretion. So it must be something new. So let's have a look in immunofluorescence. Where is EDEM? Well, EDEM is a part of it in the endoplasmic reticulum because it colocalizes with molecules that have these short sequences. This is a retention signal for ER-resident protein. For example, proteins like PDI, BIP, or careticulin have this retention sequence. So the red is the reticulum, and the green is where EDEM is. Part of it is in the reticulum, but part of it is in small vesicles. They have a diameter between 200 and 800 nanometers. And we call these vesicles edemosomes because they contain EDEM. So what's in these vesicles? So one thing that is in this vesicle is LC31. So LC31 coats vesicle containing EDEM, while LC32, which is a specific marker of autophagosome, does not coat, does not colocalize with vesicle containing EDEM. So this is another demonstration that autophagy has nothing to do with degradation of EDEM because the autophagy marker LC32 is not present in these vesicles. So LC31, when we did this study, was considered a cytosolic precursor of LC32. LC31 is in the cytosol, and when autophagy is activated, LC31 binds to the membrane of the autophagosome covalently. It covalently binds to phosphatidylethanolamine, which is a lipid in the membrane of autophagosomes. We found that LC31 is not only in the cytosol, is also associated with vesicles, these ones, that contains EDEM. We could separate this vesicle containing EDEM and LC31. This would be the mobility of LC31 and this the mobility of LC32. We could separate these vesicles from the endoplasmic reticulum with density gradients. Here you have low density and here you have high density. The endoplasmic reticulum that contains, for example, calnexin and part of the cellular EDEM, and the edemosomes that contain the majority of EDEM and LC31 are separated because the endoplasmic reticulum is lighter, has a lower density compared to the edemosomes. In this vesicle, we later on found other proteins that are rapidly degraded from the endoplasmic reticulum, other ERAD regulators of degradation. So this is not only for EDEM, it's also valid for OS9 and also valid for cell 1. Two of these chaperones that are in this part rapidly turned over. So they also enter these vesicles. So we have this model. This model claims that at steady state, you want to promote folding over degradation. To do that, you remove the proteins that regulate the degradation. And to remove this protein from the ER, you need a receptor. Because the ER is filled with huge concentration of chaperones. And the long-living chaperones are just mixed with these short-living chaperones. So you have to selectively take this out in vesicles coated with LC31, and then this vesicle fuse with an acidic compartment. So we have something like a novel vesicular transport out of the ER. So the conventional secretory pathway is this one, COP-coated vesicles that are released from the endoplasmic reticulum to the Golgi compartment. We have a sidewise separation of vesicles. And we were reasoning, okay, if this really exists, then there must be a pathogen, a virus, that exploits that hijack that. Because viruses, and here we arrive at this point, at coronaviruses, viruses are very smart. They use machinery that our cells have in place. So for example, if you take influenza virus or Semliki, an alpha virus, they bind to receptors, and then to deliver their genetic material into the infected cells, they exploit the endocytic pathway, not only to enter into the cell, but also because they find here an acidic pH that changes the conformation of the spike proteins, of this protein at the surface of the virus. This protein change their conformation, and this results in the fusion of the membrane of the virus with the membrane of the endosome, so that the RNA or the DNA of the virus is released in the cytosol. And then they use our replication and transcription, translation and folding and transport machinery to synthesize their proteins, to fold their proteins in the endoplasmic reticulum, and then to transport their protein at the surface of the cell where the progeny will bud. So they really exploit our cells. So now we are not talking about the secretory pathway, we are talking of the selective release of vesicles that do not contain calnexin, do not contain BIP, so abundant ER markers, but that contain EDEM and OS9, released from the endoplasmic reticulum and fusion to the edemosome, to the lysosomes. So is there a virus that hijacks that? So looking at the literature, obviously there is a virus because I'm telling you that. So in the literature, we found a virus, the coronavirus, so for example, the SARS virus or the mouse hepatitis virus, that in infected cells produces vesicles that buds from the endoplasmic reticulum, similar to like the edemosomes that we are studying. So they bud from the endoplasmic reticulum. And these vesicles do not contain abundant ER proteins like calnexin and BIP. There was at this time no marker in these vesicles. Nobody was able to, so being derived from the ER, people were looking for ER markers, for Golgi marker, for intermediate compartment markers. None of them was there. So it was unknown what was in these vesicles. Well, looking at that, we thought that maybe they contain EDEM. They derive, maybe are the vesicles that we are looking at. So we contacted a group, or two groups, working on coronavirus in Holland, Fulvio Reggiori and Cornelis de Haan, and we asked them, could you please try and see if in this vesicle you find EDEM? And there was EDEM inside. Then we asked, okay, could you please look if there is an SC31 coat? And they could find an SC31 coat. So what we think is that this mechanism that I am describing is hijacked by coronaviruses. Now, coronaviruses do not really need the content of the vesicles. We can deplete OS9, EDEM from the cells, and coronavirus can replicate. But we cannot deplete, for example, SC31, which is required for the formation of the vesicle. If you deplete SC31, the infection cycle of the virus is stopped. So what does that means? That the viruses need the vesicle, the membrane of the vesicle in particular, because then here the virus targets the replication and transcription complex, the viral replication and transcription complex. So they need these vesicles, and only these ones, not these ones, for example, to put them to put their RNA transcription and translation. So now we are looking for the receptor. If the model is correct, then there must be a receptor in the membrane that binds specifically EDEM and OS9 to cargos of this vesicle and does not bind ERP57 and carreticulin, for example. So to find the receptor, we were helped by the literature, as always. There is a conventional historical model of how misfolded proteins are degraded from cells. And this model predicts that there are proteins, EDEM and OS9, that modify the sugar. So this sugar is modified by the glucosyl transferase that put the glucose and folding chaperones. By eliminating this mannoses, the activity of GT is not possible anymore. And the idea is that these two proteins binds the sugar of the proteins, modify the sugar of the misfolded proteins. And then the misfolded proteins is transported at a channel in the membrane of the endoplasmic reticulum. And this channel is a complex of many proteins. It's called dislocon, because this serves to dislocate the misfolded protein into the cytosol. And here there is the proteasome that will eat the misfolded protein that has been injected into the cytosol. So this is the historical model. You have this protein that recognizes sugars in the substrate, deliver the protein, the misfolded protein to the dislocon, and then the protein is dislocated. More recently, a new model has been proposed, and in this new model, these two proteins do not bind the sugar of the chaperone. These two proteins do not bind the sugar of the misfolded proteins anymore. They bind sugars displayed on the dislocon. This is a completely different model that contradicts 20 years of studies by many groups. So this protein would bind the sugar on cell 1, and then would transfer the misfolded protein there, and the protein is degraded. So we were wondering, is that really true? So we did this experiment. We have cells in which, let's say they are steady state. They don't express a misfolded protein. They are fine. And then you look if there is this complex. So we immunoprecipitate HRD1, and then we make a blot, and we stain with an antibody for cell 1. And as you can see here, in normal cells, there is this amount of complex. So these proteins are in complex. Then we put a misfolded protein. So under this condition, the degradation machinery is active because it has to degrade this protein which is misfolded. And what happens is that the content of this complex, which is active in degradation because it has to dislocate the proteins, the content of this complex in the cell increases. If we put the same amount of folding protein, this is not misfolded, this can be secreted, the amount of complex goes back to normal. So the amount of this complex involved in degradation is increasing when the degradation machinery is active, but is lower when the degradation machinery is inactive. So kind of at steady state, you kind of disassemble, but then when you need, you really assemble the complex. It's more stable. What happens with the complex between EDEM and cell? So people were saying, okay, EDEM is binding to cell, and this is involved in degradation of chaperones, because then EDEM discharges substrate here. So it's kind of opposite. When the degradation machinery is inactive, you have a lot of this complex. Here we have immunoprecipitated EDEM, and then we have done a blot with cell. When we activate the ERAD machinery, the complex is disassembled. So now we need to destroy proteins, but the complex is not there. Then we put a folding competent protein, and the complex is formed again. As if this complex is not formed when you need degradation of substrate. Is formed when you don't need degradation of substrate. And when you don't need the degradation of chaperones of substrate at steady state, you want to remove this from the endoplasmic reticulum. At steady state, you want to promote folding. So you want to lower the concentration of that. So we were wondering, maybe the complexes that people are saying between cell and EDEM is the complex that we are looking for, is the complex that brings EDEM and OS9 into the vesicle for degradation in the absence of ERAD activity. So this is what happens. So we have the information that cell 1 associates with EDEM and OS9. This is fine. Could be a receptor. We have another information. This association is improved at steady state when cells want to remove this chaperone. So could be the receptor. Now there are three predictions. The first one is that we should find cell there. If this is the receptor, it should go with the cargo in the transport vesicle. The second prediction is that, well, cell is a transmembrane protein. Maybe cell binds the coat. So maybe we can identify a complex between cell 1 and SC31, which is the coat of the vesicles here. And the third prediction is that if we delete this receptor, we should stabilize EDEM. EDEM is not transported outside. This is what I will show you in the next 3 slides. So the edemosomes, so the small vesicles that contain EDEM, do not have markers of the endoplasmic reticulum. They contain EDEM, they contain OS9, and in density gradients, they are denser than the endoplasmic reticulum. So now, where is cell? Well, cell 1, exactly like EDEM, partially colocalizes with the endoplasmic reticulum marker KDL, but it is also found in spots, small vesicles of 200, 800 nanometers. And if you look in these spots, well, in these spots, you don't have only cell 1, but you also have EDEM. So these spots are yellow because they contain EDEM and cell 1. And if you look with another marker, like the spots that contain EDEM, also the spots that contain cell are coated with LC31. So they contain both cell and LC31. If you separate the vesicles in gradients, you have the endoplasmic reticulum. Here, PDI is a conventional endoplasmic reticulum marker that migrates in this fraction of the gradient, while the proteins that are rapidly degraded certainly are found in the endoplasmic reticulum, but then are also found in denser vesicles that are coated with LC31, that are co-stained with LC31, that do not contain ER markers, but that contain OS9, EDEM, and cell 1. So cell 1 is in the same compartment. So now, does cell 1 bind LC31, which is the coat? So to look at that, we have precipitated from cells the endogenous cell 1 and the endogenous calnexin. Calnexin is a protein like cell 1, is a type 1 protein with a long cytosolic tail. The same is for cell 1. So if we precipitate cell 1 and then we make a blot to look at the presence of LC31 in the precipitate, we see that LC31 co-precipitate with cell 1. If we do the same with calnexin, we don't have LC31 that co-precipitates. So cell 1 binds LC31, calnexin, which is an ER protein with the transmembrane region, doesn't bind. Then we have expressed recombinantly these proteins to look more carefully if the tail of cell 1 really binds LC31. So the recombinant cell 1 binds LC31 because when we precipitate cell 1, we make a blot with LC3 and LC3 is there. But if we put the tail of calnexin, we replace the tail of cell 1 with the tail of calnexin, now we precipitate the chimera, we don't have, or we have a lower amount of LC31 that co-associates. And we can do the same with calnexin. Calnexin, ectopically expressed, precipitate a low amount of LC31. But if we put the tail of cell 1, then the amount of LC31 precipitating is much higher. So we consider that background level, we consider that a significant co-precipitation that shows that cell 1, or more precisely the tail of cell 1, binds LC31. If we express recombinantly cell 1 in the cell, we have cell 1 in the endoplasmic reticulum, is the red, but then we have recombinant cell 1 also in vesicles that co-stain with LC31. This is another way to show that cell 1 and LC31 form a complex. So the recombinant cell 1 exits the endoplasmic reticulum and ends up in vesicles that are coated with LC31. If you do the same with cell 1, in which the tail has been replaced with the tail of calnexin, well, LC3 is still in these vesicles, but now you don't have cell 1, because the tail of calnexin prevents the movement of cell 1 from the endoplasmic reticulum into these organelles. Calnexin is not going into this vesicle stained with LC31, it remains in the endoplasmic reticulum. But if you put the cytosolic tail of cell 1 to calnexin, now this hybrid molecule goes into these vesicles that are stained with LC31. So the last question was, okay, cell 1 is the receptor. It binds to EDEM. It binds also to LC3. It promotes formation of these vesicles. But then if we remove that from cells, we should stabilize EDEM. EDEM should remain in the endoplasmic reticulum. And this is what happens. So in this experiment, we have specifically downregulated LC31, or we have specifically downregulated LC3, or we have specifically downregulated cell 1. And if you look at the sample in which cell 1 has been downregulated, you will see that there is more EDEM compared to control cells. There is more OS9 compared to control cells. But if you look at a long-living chaperone, you could look at BIP, GRP94, calnexin, the level of this chaperone does not change. What happens is that if you remove cell 1, you slightly decrease the turnover of EDEM. The effect is not dramatic, but there is a stabilization of EDEM. So here is the degradation of EDEM in normal cells, and here is the degradation of EDEM in cells that have a low level of cell 1. These are not knockout, these are knocked down, so there is a residual amount of cell. Here is the situation for OS9. The half-life of OS9 is like this in normal cells, while if one removes cell 1, the protein is stabilized. So the fact that you have more proteins when you delete cell 1L is due to the fact that you stabilize the protein, you delay the degradation of the protein. So to conclude, what I showed you is that in our opinion, this protein is the receptor that specifically extracts ERAD factors but not folding chaperones from the endoplasmic reticulum for disposal. So this is what I showed you today. I hope that it's a bit more clear what this concepts represents. I showed you that the endoplasmic reticulum is a very important organelle for production of protein. It contains a folding machinery, a quality control machinery, which is very nice because our protein can productively fold. But it also contains a degradation machinery. This could be dangerous because the degradation machinery, if the level would not be regulated, could compete with the folding machinery for nascent chains. So we think that at steady state, cells modulate the activity of the ERAD. ERAD tuning means tune downs the level of degradation machinery to promote, to make so that nascent chains can fold efficiently. So I have to thank especially Riccardo Bernasconi and Carmela Galli for this work, and then Fulvio Reggiori and Cornelis Dehaene for the work with the viruses that hijack this pathway for their replication. Thank you.